Histone tails in nucleosomes play important roles in gene access and regulation of many biological processes, including DNA transcription and chromatin compaction through post-translational modifications. In this video, we will be looking at the role of histone tails in histone-DNA interactions. So first, how exactly is DNA packaged as chromatin? It turns out DNA is packaged with the help of histones. A histone octamer consists of two copies of each of the core histones, H2A, H2B, H3, and H4. These histones are made out of mostly positively charged residues, such as arginine and lysine. The positive charges allow them to interact and bind to the phosphate backbone of DNA that is negatively charged and form what is called a nucleosome. So now the DNA is able to wrap around the histone octamer and become more tightly packed, because the phosphate backbone in the DNA has been neutralized, and between the two nucleosomes, they are interconnected by a short linker DNA. So we know that the histone tails are critical for modulating various biological processes. But how does each tail affect the structure and stability of the nucleosome? Well, in a study by Iwasaki and colleagues, they first determined the crystal structures of 4 mutant nucleosomes, where each nucleosome lacked the N-terminal tail of one of the 4 histones. So here, this is showing a top-down view of a wild-type nucleosome with N and C terminal tails labeled in 4 different colors. The helical DNA in black wraps around the colorful histone octamer in the middle, and in the right panels are the 2 closed-up views of the N-terminal regions of H2B and H3. Interestingly, Iwasaki and colleagues found that the deletion of H2B or H3 N-terminal tails caused ambiguity in the electron density map compared to the wild-type nucleosome. You can also see that some hydrogen bonds, shown in red, are disrupted or weakened between the DNA in white and the H2B or H3 N-terminal region in blue. So based on this study, the deletion of H2B or H3 N-terminal tail disrupts histone DNA context, and thus substantially reduces its stability due to the disruption of hydrogen bonds. In another similar study by Fu and colleagues, they examined the effect of lysineacetylation on the H2B N-terminal tail in lesion-free nucleosomes. It was found that in the lesion-free unacetylated nucleosome, the H2B tail, shown in red, is collapsed onto the DNA surface and forms an extended conformation. This enhances nucleosome stability and allows favorable electrostatic interactions between the negatively charged DNA phosphate backbone and the majority of positively charged arginine and lysine in the histone tail. Now, if you look at the lesion-free acetylated nucleosome, the tail forms a compact conformation and is released from the DNA surface, which makes it more solvent exposed and dynamic. This reduces the electrostatic histone DNA interactions as well as the contact between the tail and the DNA. Both of these studies investigated the precise contribution of each histone tail, where they are required for the stability of histone DNA interactions. These findings are crucial information for understanding the complex roles of histone tails in modulating chromatin structure and cellular functions.